Hello, friends of astrobiology. Welcome to our brand new show on SegaNet, Ask an Astrobiologist. It replaces our previous show, Talk to an Astrobiologist. And the reason we're upgrading our format a little bit is we got new resources from the NASA Astrobiology program, as well as ELSI, the Earth Life Science Institute at Tokyo Tech, and the nonprofit Blue Marble Space to make this a little bit more dynamic and hopefully a bit more interactive and informational for you all. So if you have any questions during the program, make sure you use the hashtag AskAstrobio to post your questions online, or use the SegaNet chat if you are on seganet.org. So it's my great pleasure to invite back to SegaNet Dr. Charles Cockell. Charles is a professor of astrobiology, one of the few in the world who holds this title at the University of Edinburgh in Scotland. He's also the director of the UK Centre for Astrobiology and all-around baller astrobiologist. If you have not taken his course on Coursera on an introduction to astrobiology, I highly recommend it. Charles, well, are you wearing a mining suit? Did you just spend the last few hours 1 kilometer below the surface of the Earth?
It's good to talk to you again, Sanjoy, and that's exactly where I've been. Actually, by coincidence, this interview was like 20 minutes after I came up from our underground astrobiology lab in the Bulby mine, where we've been studying life deep in 250 million year old Permian salt deposits. So we've been down there today doing some science, collecting microbes, filming with the National Geographic, and then I literally got up 20 minutes before this interview, so that's why I'm in an orange mining suit. So I'm not actually in prison. I've just been down the mine collecting some samples, doing some filming and working in our lab, which I should say is actually was the first astrobiology lab to be built underground. So it's an exciting facility that we have.
That's so cool. So the Permian is about 250 million years ago. So let's go a bit before that and tell us a little bit about your childhood.
So it's 250 million year old salt deposits. It's very old, but it's not as old, say, as early Mars. But it is an environment where we can look at microbes living in salt environments and try and understand how they live in different types of brines. So in the mine, we've got sulfate brines, we've got sodium chloride brines, all the sorts of salts that you see on Mars. So it's a really interesting place to look at microbial communities in extreme briny environments and understand how they adapt to those extremes. And it might tell us something about the sort of adaptations and functional capabilities of microbes living in briny environments on early Mars.
Yeah, and brines are also really important for thinking about life on Europa. But I want to go back a little bit before that, because you're an astrobiologist today, but when you were a kid, when you were growing up, I would like to learn a little bit more about what events made you become a scientist and how you ended up finding the astrobiology field and choosing that as a career.
Okay, well, I'll try and keep it brief because it's a long story. I actually began, I was interested in natural history from a very young age. So I used to collect moths and insects and things. And then when I went to school, I couldn't decide between biochemistry or astrophysics. And I decided in the end to do biochemistry. And that's what I did my first degree in. Then I got a fascination in genetic engineering and did medical molecular biology for my PhD at Oxford. So I actually studied the proteins involved in blood clotting. But that didn't really satisfy me. I had an interest in space exploration, astrobiology. So I spent one summer during my PhD at the International Space University, where I met what would become future colleagues at NASA Ames Research Center. And then I got a postdoctoral position at a National Research Council associateship at NASA Ames and went out and did a postdoc. And that's really when I became a sort of proper astrobiologist, if you like. I spent 4.5 years at NASA, and then I came back to the UK and worked with the British Antarctic Survey. And then I spent 4 years working on Antarctic research, and then I got my professorship in astrobiology at the Open University. So my career has sort of taken different directions, from biochemistry to molecular biology to astrobiology, but all the time I've always tried to link biology with space exploration, and that's always the thing that's fascinated me. And now I find myself as a a professor of astrobiology doing it full time. It's been my sort of childhood dream of a job, so I'm very lucky, I think.
Space, but yet spend a day 1 kilometer below the Earth's surface, which is pretty awesome. So, but the path you've taken, I'm sure, was not a straightforward one. I was wondering if you could tell us a little bit about like the hurdles you encountered and the mentors you met who kind of helped you along the way to become the amazing scientist you are today.
Well, I'll defer on the amazing scientist. I think it's for other people to make a judgment, but I can tell you about the hurdles I crossed to get where I am. You know, one of the most difficult things I think is funding and building a career. People see you as a professor and they think, oh, you know, he must have had an easy time or got there easily. You know, I went out to NASA and then ran out of money, and I had to go and scrounge for money for 8 months from Stanford, and then I ran out of money there and went down to the University of Arizona, where I then ran out of money there, and then I went back up to Ames and ran out of money there, and then went to the British Antarctic Survey for 2 years when I first came back to the UK to do research here, and then I ran out of money after 2 years. It's not an easy thing, and it's taken a lot of sort of times of difficulty and uncertainty with research positions, and that's not to depress people. It's more to give people hope that you just have to keep going and be persistent. It's not actually easy for anyone developing a career in science, but if you believe strongly enough in what you're doing and keep going, things tend to work themselves out. But you know, it just takes focus and a lot of work. My advice is write papers. You know, I know it sounds like a simple piece of advice, but being productive is one of the most important things for achieving a permanent position and getting a job at a university. As long as you are productive, you demonstrate that as a scientist you can get information out there. So I think that's an important thing. Persistence, being enthusiastic, believing in the dream that you're pursuing, but at the same time also getting those papers written and being productive, and you can't really beat that combination of things.
I echo those sentiments, and I agree. It's definitely, as a personal scientist myself, I definitely appreciate the challenges of obtaining money to do science. Could you tell us a little bit about the mentors you've had throughout your career and the advice of wise people you've taken and that have helped you guide your career?
Yeah, I mean, I've had a lot of, I've been lucky. A lot of people helped me out. You know, people at Ames, Chris McKay, Lynn Rothschild, others who supported my career when I became an astrobiologist. Without their support, this wouldn't have happened. I should also say not just mentors, but colleagues as well, people I've worked with at Ames when I was a postdoc fellow scientists who were supportive as well. I had a lot of support from senior people. Barry Bloomberg, who was the first director of the NASA Astrobiology Institute and Nobel Prize winner, you know, wrote me letters of support for my jobs after I finished at NASA. And, you know, it's not necessary, but it doesn't help. It doesn't hurt, rather, to have Nobel Prize winners writing reference letters for jobs. But, you know, people listening shouldn't think you need that. But I guess it is an illustration that it is important to you know, to work with senior people and be positive towards your seniors, whatever whatever they might tell you. And that's certainly part of the help that you need to develop a job. So I've been lucky. A lot of people have been very supportive of what I wanted to do in life and have supported me at critical times. So, you know, I have little hesitation in saying that the papers and the dream and the vision is important, but it is also important to try and get to know people who can help you out at critical times.
Yeah, I don't think it's luck, Charles. I mean, you're very good at communicating your enthusiasm, and it's pretty contagious to the people around you. So give yourself some credit there.
Yeah, I mean, there's hard work. You don't get anything for free in life, and it can always look like to other people you've been lucky, and very little of it is luck. It's hard work and yeah, persistence. But the good news is that we live in a world these days where persistence and hard work does actually pay off. I think a few centuries ago, it was much more about who you knew, and there was a lot of luck. And that's a good thing, that if you believe in what you're doing and people want to be astrobiologists, if you show that persistence, you have a high chance of getting what you want. So you just have to keep pushing on.
That's excellent advice. One thing I really like about you, Charles, is you're a very bold scientist, and the experiments you carry out are just really awesome. And I was wondering if you could tell us a little bit about the experiments you actually flew into space.
Yeah, so we have done space experiments to International Space Station. I really want to go to the Moon and Mars, so I should say that this is really just the first step. But I'm not going to complain anyway. But we've been lucky. We've had European Space Station experiments, where space agency experiments, one of which was quite fun. We took some rocks from a cliff in the south of England, in a place called Devon, a fishing village. We took the rocks and we flew them on the outside of the International Space Station for a year and a half to ask a very simple science experiment, which was a science question, which was to ask, did any microbes survive? And that year and a half in space, and one species survived, and we have that growing in our lab now, and we're trying to understand how it survived in space. And two of the answers are that it forms biofilms, and that it's also very resistant to ionizing radiation. So that was really the first experiment that I did on space station. Currently, we're preparing another experiment that's going to fly in 2 years, which I'm PI of, called BioRock. And it's to look at how microbes break down rocks in microgravity and simulated Martian gravity using the cubic centrifuge on the space station. And this is all about growing biofilms in regolith on Mars and on the Moon, and whether we can use microbes to break down rocks, produce soils, and maybe even do things like biomining, so extracting useful elements from rocks like iron and titanium and precious metals like platinum from asteroids, and use microbes to do this work for us, just as microbes are used on the Earth to do biomining. So this is a really exciting experiment. It's leading to a new generation of microbial growth reactors that we're building with Kaiser Space, who are our industrial collaborators. And what we hope to do in the next 3 or 4 years is build tiny little cubic microbial growth chambers that you can fly on CubeSats or on space stations or send to the Moon, in which you can do microbial growth experiments, microbial culture experiments. So these are little modularized culture chambers, if you like, that you can use in space. So that's another one of our sort of exciting space experiments.
So with those microbes that do biomining, I think that's really interesting, because I mean, one of the big push from the private industry these days is to do asteroid mining. But is the kinetics of the microbe breakdown fast enough? Is it scalable? It seems like it'll be good for a small project, but on the larger scale.
Well, about 20% or so of all the copper on the Earth today is extracted from rocks using biomining. So this is not speculation. This is a massive economic enterprise on the Earth. And yes, so microbes accelerate. You can think of them simply as catalysts for accelerating, say, the oxidation of iron or sulfur into sulfuric acid and acidifying the rock environment to break down rocks. So microbes can catalyze chemical reactions orders of magnitude, sometimes up to 1 million times faster than just purely chemical reactions. And that general ability of microbes to catalyze reactions is at the heart of biomining, whether that's specifically extracting particular metals from rocks, or just using microbes to help break down the silicate matrix of rocks and release economically useful elements. So yes, microbes can do this on a big scale. How you would do that on an asteroid or on the Moon or Mars, when on the Earth these microbes generally do this in aerobic environments where they've got plenty of oxygen. So you'd have to get the oxygen, and that's an energy and mass cost to do all that. So scaling up biomining to extraterrestrial environments is at the moment something we don't fully understand whether it would be economically scalable. I think that's yet to be demonstrated. But it is just one of the ways in which we could use microbes, use organisms to do things for us in space that might improve our ability to get access to resources or ameliorate soils, or even do things like use microbes for extraterrestrial gardening, something as trivial as growing plants and microbial mats in stations on the Moon and Mars. Microbes can do that work.
If we were to think about the long-term human spaceflight, we'll definitely need to have an understanding of how microbes work in a space environment. So it's kind of cool that this is like one of many examples of practical astrobiology, and you can't talk about geology without talking about biology and vice versa. So it's a very, very exciting field. You're also conducting, if I remember right, one of the longest running astrobiology experiments, right? You started something, an experiment's going to last like 500
Yeah.
years or something. Could you tell us
Yes.
about that?
Yeah, sure. I was having a conversation with some of my students in my lab, and we were saying, what is the mathematical function that describes the loss of viability in a desiccated microbe? Whether that's a microbe that's dried on the surface of Mars or a microbe in permafrost on Earth, what is it that killed the microbe over long time periods? Is it accumulated DNA damage, or is it just damage to the cell membrane over a short period of time? And we looked around, and of course we found papers where people had looked at survival of desiccation of microbes. So there's a paper about nostoc, which is a cyanobacterium surviving for over 100 years. That was some herbarium specimens that people had got out of a botanical collection. But all these papers are quite haphazard. They're samples that people have found in museums or in other places. And we thought, why don't we do the experiment properly and set up a 500-year microbiology experiment? So that's what we've done. What we've done is we've sealed up Crocokyriopsis, which is a desiccation-resistant cyanobacterium, and spores of Bacillus subtilis. So we've got a vegetative and a spore-forming microbe. We've sealed them in glass vials. So what will happen is we take a triplicate vial every 2 years for the next 25 years, and then every 25 years for the next 475 years. And the experiment ends on June the 30th, 2514. And we've got in the experimental box, we've got vials that are just exposed to the environment, that are just in the box. And then we also have vials inside a lead box to cut down some of the background radiation, to test the hypothesis that background radiation is one factor in loss of viability. And then the entire experiment is reproduced. So one of these experiments is in Edinburgh, and we have a complete replicate 500-year experiment that's being curated by the Natural History Museum in London to ensure there's redundancy, but it also means we get 2 data sets from 2 different environments. So we've got biological triplicates in each experiment, and then the whole experiment is technically replicated in 2 institutes. And we took the first time point this year, so the experiment began 2 years ago. July this year was time point 1, which was a matter of great excitement in my lab. And I can tell you now that neither organism significantly lost any viability in both the exposed and in the lead boxes. That's good news for a number of reasons, not because it tells us much. We know that spores will survive 2 years. That's a short time period. We know that Crocokyriopsis will survive for 2 years. But what it has shown us is that the preservation mechanisms, the preservation method that we use for the microbes has worked. And so we have an experiment now. After 2 years, there's no loss of viability. So now the question is, what will happen over the next 500 years? Will we see a sudden crash in viability, maybe over the next few years as those organisms die? Or will we see a crash in viability, say, in 150 years when there's a certain accumulated level of DNA damage? And what is the difference between a vegetative cell and a spore-forming cell? And then even if the cells die, let's say in 100 years they're all dead, we can still follow the destruction of biosignatures over 500 years, what happens to the membranes and the DNA and nucleic acids or proteins. So this experiment provides us with an understanding of the degradation of biological macromolecules over century timescales. So we're very excited about it.
I can tell. Does it have applications for life on Mars?
I think it does. I mean, of course, Mars has a much higher radiation exposure on the surface, but it might tell us something about life underground that's desiccated in a dormant state and what the processes are of loss of viability. I mean, I should say 500 years is still a short experiment on a microbial timescale. I mean, microbes probably can survive millennia in permafrost or completely desiccated. But the purpose of the experiment was to try and get to timescales that are more reflective of microbial timescales than human timescales.
That's very cool. I would like to change gears a little bit here. So you're a professor, you're director of the UK Centre for Astrobiology. You do all this amazing science, and yet you had time to write a book, an astrobiology textbook for that matter, for undergraduates. Where do you find the time? How do you do it? Like, tell us more about the process that led to this book.
Okay, well, when I went to Edinburgh about 5 years ago. That's when I set up the centre, and of course I need to teach. And one of the things I wanted to teach was astrobiology. So after a couple of years of teaching physics, just to get to know the undergraduates and how the university works, I set up an astrobiology course at Edinburgh that's been very popular. It attracts students from 11 different departments across the university, from sciences. And as part of that lecture course, I needed to write lecture notes for my students to go with the lectures. So I wrote all my lecture notes, and at the end of the first year of teaching, I looked at all my notes and thought, hang on, this is 50% of a textbook. Why don't I just write the other 50%, and then I can publish a textbook, and then I won't have to write lecture notes. I can just tell my students to go and buy the book in the bookshop. So, you know, at the end of the day, it is actually a time-saving move to write a textbook, because it'll be updated by Wiley, hopefully, and then it can be used in other universities. But actually, slightly more seriously, the other reason for doing it is I thought, I've got all this experience of teaching astrobiology in a university, and I know now which lectures work and which material students found exciting. It just seemed like a really good idea to write a textbook that could be used in other universities to save other people the time of having to reinvent astrobiology courses and figure out all the things that I've taken 3 years to figure out with what sort of lectures to teach in mind. I should also say that I wrote 21 lectures in PowerPoint slides that you can get on the Wiley website for free, and they go alongside the lecture notes, and they're based on my own lectures. So if anyone out there wants to set up an astrobiology course at a university, you can download 21 lectures that have 50 minutes worth of PowerPoints, and then you use the textbook and you have an instant astrobiology course. So another motive for writing this textbook was to try and encourage people to set up more astrobiology courses in universities around the world. And, you know, as you know, the more astrobiology courses there are, the better for all of us. It just means there's more astrobiologists, more support for astrobiology. So in summary, the reason why I did it is because I'd written 50% of a textbook already. It seemed like a small step to make a big impact in improving astrobiology around the world in universities. That's why I wrote it. And it's fun to write. I like writing. So, you know, this was not a chore for me. It was something fun to do.
Good for you, Charles. And I read a few chapters, and it's very easy to read and a very well designed textbook. So good job. So before we open it up to the public, because we've been talking for roughly half an hour, I have one more thing I'd like to ask you. At the mine you're at now, Build Me Mine, you're organizing a course next year, right, based on NASA Spaceward Bound.
Yeah.
Could you tell us a bit more? When is it going to be held? What it's about?
Yeah, it's in October. It's a collaboration between the Bulby Underground Science Facility here, where we're doing astrobiology, and NASA Spaceward Bound. And it's to test instrumentation, to do science in a deep subsurface environment, and also to do education and outreach in the mine, a kilometer underground, although people can also work on the surface and do outreach activities here. We've put out a general call that you can find on the UK Centre for Astrobiology website, or you can contact me directly. My email's on my website. And we're inviting anyone with really good science, good technology, or good outreach that involves understanding life deep underground, testing for the subsurface exploration of the moon or Mars, or just doing good outreach in astrobiology and underground science to take part in this. And then what we'll do is we'll look at all the proposals and come up with a program. So it's October next year, the 9th to the 24th, I think. I've completely forgotten the dates. I think it's midway through October. It's 2 weeks. It's in Whitby, just in Yorkshire. The mine is just north of Whitby. It's great fun as well. It's a fantastic environment to work with, and you will meet other people working in astrobiology and outreach. We already have JPL involved, so they're going to be here testing Mars 2020 instruments. So it's going to be a really fantastic 2 weeks. Anyone interested in joining us, please get in contact and send us in all it takes is a half-page proposal on your experiments and what you plan to do. And then you can wear one of these very stylish orange jumpsuits like I'm wearing now.
That's great. And as an alumni of 2 NASA Spaceward Bound programs, I love the combination of science and outreach. That's just a testament to the program. It's not just science, it's not just outreach, it's both, and both thoroughly, which is awesome. So exactly. Definitely those of you who are excited about those 2 topics, science and outreach together, really consider Charles' Spaceward Bound program next October. So okay, so sums up all the questions I had. I am scanning the Twitter feed right now and the SegaNet chat to see if there are any questions. There's one from JP Heavy Industries at JP Heavy who asks, what would a methane-based ecology look like on, say, Titan?
Well, that's interesting. I mean, Titan has a bit of a problem because it's at about 94 Kelvin, which is some way below what we think is the lower temperature limit for life. So, you know, certainly there's the raw material for life there. Lots of organic materials, complex organic materials, an interesting photochemical cycle that's generating complex organics, the sort of things you might build living things from, although we haven't yet detected things like amino acids and sugars. That's yet to be demonstrated. But the real problem is can you have life working at 94 Kelvin? There have been some interesting ideas about life on Titan. So, for example, ideas from Chris McKay and Heather Smith at NASA Ames, who proposed life using acetylene and hydrogen as an energy source. So there are molecules as well on Titan that might be plausible reduction oxidation reactions for getting energy. But I think still the problem remains, what's the solvent? Liquid methane maybe, but liquid methane has some disadvantages with respect to dissolving polar substances and doing the sorts of things that we associate with biochemistry. And I think the real problem is low temperature. I mean, okay, you might say maybe life is just working very, very slowly there at literally glacial timescales, and you know, organisms just divide every few 100 million years. But it's very difficult to get reasonable kinetics going at those temperatures, where you can conceive of an organism that can adapt fast enough to changes in environmental conditions that we associate even with environments like Titan. Titan is a dynamic world despite the low temperatures. Can you have an organism that can adapt to those changes quick enough when it's operating at 94 Kelvin? I think that's an interesting question to ask.
Yeah, it's a bit warmer underground. There is a liquid ocean, but it's between ice. It's not contacting with rock, so I'm still not convinced that it's a great place for life.
Absolutely. Maybe that's what the questioner was asking. So yes, I mean, if there's a subsurface ocean, that would be another question altogether. And if there's liquid water, even maybe liquid water and ammonia, a mixed ocean, that might be a more plausible environment for life than the near-surface environment.
Yeah, no, agreed. Let's see, what else is on the Twitter feed, on the SegaNet chat? Ben Pierce asks, where will we find the first evidence of life beyond Earth? Will it be Mars, Europa, maybe an exoplanet?
Well, this is always, you know, I mean, my guess is as good as anyone's. Where do I know where life is? But it's an interesting question because, you know, there is a possibility that we might find life on an exoplanet before we find life in our own solar system. Unless there's life in Enceladus and Europa that's being spewed out into space in plumes, and that might be relatively easy to capture. But, you know, organizing a sample return mission or a life detection mission to Europa and Enceladus to sample those plumes may well be a decade or more in the future. Finding evidence for life on Mars might require drilling into the subsurface. That could be a decade or a few decades ahead to really get good samples out that unequivocally show life. Within that period of a couple of decades, it's not impossible that we might be able to find an oxygen biosignature on an exoplanet. So I think, you know, it would be remarkable in astrobiology if we actually found evidence of life beyond our own solar system before we found life in our own solar system. But who knows? I mean, we may find we're in a biological desert and that all Earth-like exoplanets have no life, in which case we're back to searching for life in our own solar system. So my guess as to where we'll find life is no better than anyone else's guess. I don't know, but what's really exciting for astrobiologists is the discovery of liquid water environments in our own solar system and beyond. Discovery of super Earths that may be ocean worlds, the discovery of liquid water coming out of Enceladus, liquid water in Europa, extraordinary evidence for long-lived water bodies and particularly sediments on the surface of Mars, incredible images from Curiosity of sedimentary layers. All of these things provide us with the impetus to search for life, regardless of whether we actually find it or not. So we've got the point, I think my summary would be, we've got plenty of places to look for it, and that's the good news.
What does your intuition lead you to in terms of where in our solar system you think life is? Just gut feeling.
You know, it
Tough
depends.
question, sorry.
It depends what time I woke up in the morning and what I ate that day. You know, sometimes I look at pictures of sedimentary layers taken by Curiosity in a volcanic environment that looks so like Iceland, and you just think, is it really the case that all of those sediments that Curiosity is studying were completely sterile? And I find it difficult to believe that they are. And then another day, you know, I'll see a news story about plumes on Enceladus, and I'll just think that's incredible that you have, you know, large water bodies, you know, in the case of Europa, that have 2 times as much liquid water as the Earth's oceans combined. And you think, how can that be completely sterile? But maybe these places are. I mean, it's very difficult for us on the Earth to accept the idea of large bodies of liquid water that are habitable but completely sterile. And that's a paradigm that's very difficult that's very alien to ecologists and to most of us as biologists. But it's possible that if these places never had an origin of life, or life was never successfully transferred to them from the Earth, then they may be sterile. So I don't know. I go from optimism to less optimism depending on what I've thought about and what papers I've read. But you know, that excitement of astrobiology, the fact that there are these incredible environments that have liquid water that look like plausible locations for life. Are they inhabited or not? You know, that's what we all really want to find out.
Yeah, it's definitely good for our civilization to go and find out. I think the answer to that question will have not only very profound scientific implications, but also philosophical ones as well. That would be good for us to think about. So Graham Lau, hi Graham, has a question. I think it's a very good one. So you taught on the Coursera, you've reached many thousands of people with your class, but his questions is more, how can we, I guess, share astrobiology in a more hands-on manner than just listening to lectures online? What are your thoughts on that?
Yeah, I think that's a really good question. I think there's a huge amount of potential for theory. I mean, in some sense, there is some out there, like, for example, the citizen science exoplanet projects, looking for exoplanets in data that's been sent back by Kepler and being produced by other. There are projects where there's a possibility of real hands-on projects. I think looking at the Mars Curiosity images and orbital images is something that can be handed over to citizen science. It would be great to come up with more lab-based experiments. There are people currently trying to build desktop Mars chambers that you can make by 3D printing. I had a conversation with people who are building 3D printed Mars simulation chambers. So that I think, again, with 3D printing, maybe there are planetary simulation chambers that people can build at home and do real astrobiology type experiments. But yeah, I think his question is a really good one. I think if anyone out there has ideas for hands-on astrobiology experiments, either through citizen science type projects or things that people can buy and do experiments in their own homes, that would be a fantastic avenue for increasing the outreach of astrobiology on a more sort of popular level, but also giving people who are not professional astrobiologists the chance to do real experiments.
Yeah, so astrobiology is definitely still a young field that needs a lot of ideas from the early career community and how to make it grow. There's a question on asteroid mining. Sorry, there's a question on asteroid mining by Elizabeth Frank. Hi, Elizabeth. Regarding, are you the first one to do microbes on asteroids in terms of biomining, or is there an established literature on that you're building on?
No, there's a couple of papers out there where people have looked at microbes growing on meteoritic material in the lab. So there's a couple of papers by Mortner who grew plants actually as well as microbes on chondritic carbonaceous chondrites. So it's not the first time anyone's looked at whether things will grow on meteorites, and things will grow on meteorites. Meteorites are just silicate rocks, and there's plenty of good stuff in there for things to grow in. I think ours is the first experiment that has been approved for space flight. Certainly the first experiment proposed for a space station that is deliberately focused on biomining and the use of microbes to break down rocks and play a role in biomining and also in other microbially mediated sort of reactions and useful things we can do with regolith on other planetary bodies.
That's fascinating. We have another question from Penny Boston. Hi, Penny. She asks, so biofilms are so central to the microbial way of life here on Earth. Do you think that a biofilm lifestyle is going to be a lifestyle choice of microbes everywhere in the galaxy?
It's a really good question. So hi, Penny, by the way. Good. I'm really glad you could make it. Yeah, I think so. And as Penny knows, you know, these things, biofilms are everywhere in extreme environments, in caves and other situations. Very few microbes in the natural world live alone. I mean, we call these things unicellular organisms, but very few microbes are truly unicellular. That's generally an artifact of the laboratory. When you go into natural environments, microbes cooperate in a whole variety of ways. Either microbes' energy source is another microbe's waste. They collaborate in terms of extracting nutrients from rocks. You know, one microbe might excrete an acid that will break down rocks that helps the whole community to grow. And they cycle elements as well: carbon, sulfur, nitrogen. All these elements cycle through biofilms, through different types of microbes. I think on any planet where you have microbes, there has to be a very strong evolutionary selection pressure for those microbes to start cooperating in the Darwinian struggle for survival, in the classical view of that. It's sort of biologically inconceivable you would have microbes just sitting on a planetary surface, just doing their own thing, completely separated. Because somewhere in that mixture of microbes, there's going to be a selective advantage for two microbes to work together, even, as I say, if it's just one microbe using another microbe's waste as an energy source. So I think there has to be a drive towards biofilms. I think on any planet where you had microbes, you would find things growing in biofilms.
Great question, Penny. Thanks. Graham has another one. Good question again. Kind of tickle the brain here. If you are allowed to say one sentence on a future golden record that will be sent to space, that kind of summarizes humanity almost, what would you say? Or in addition to that, what would you put on that record?
My message would be, we do good things as well, you know.
Indeed.
Sorry, that's a very cynical reply. You know, if an alien species was watching what we get up to, I think they would be in despair. But you know, we do a lot. There's a sort of serious side to that comment. Astrobiology, the search for life beyond the Earth, for me at least, is one of the things that demonstrates the best in our civilization. And I'm not just saying that because I want funding. I really mean that. Asking the question of are we alone in the universe, I don't know of any other species on the Earth that asks that question. A lot of species attack each other. They do warfare, obviously in a more rudimentary way than us. But a lot of the things that we do that are bad are products of animal instinct. But asking questions like what's the universe made of? Is there life out there? These are questions that really define us as a species that can think, as a civilization that can go beyond purely animal instincts. So I would like to think that these are things that we would want to tell other species in the universe, that yeah, we have our failings and we have our faults, and if you're observing our planet, you probably see some quite bad stuff going on. But we also think about these higher level questions. There really is hope for our civilization to break beyond violence and destruction, and to move out into the universe and contribute in a very positive way if there are other species out there. So that would be my message to other species, and maybe they would recognize that. You know, if they are species at the end of a food chain, maybe they understand aggression as an evolutionary product. And a comment like that, after all, or we actually do good stuff as well, they might actually understand that and understand the conflict in ourselves and in our civilization, and the work that we have to do to try and reach beyond destruction to the positive side of our natures. I think all intelligent species have that conflict. That's
some beautiful words, Charles. Did you get that, Graham? Yeah, I agree with you that astrobiology is a very unifying as a discipline because, looking up at the stars and wondering what's out there is definitely not something that's limited to individuals, pockets on the Earth. Everybody has done that, and it makes for wonderful conversations. And the fact that we can tackle that scientifically in this early 21st century is an awesome time to be alive.
Yeah.
So astrobiology is a fairly young field, as you know. What do you think are the challenges it's going to face in the future? Like, there's no yet Department of Astrobiology, and personally, I think that's probably a good thing. But how do you see the field evolving and maturing, I guess?
Yeah, I mean, I'm not the first one to say that, but I agree that one of astrobiology's biggest challenges is that it's so broad, it's difficult to condense it into a single field. When you can put up a building and say that's the department of X, Y, Z, it's a lot easier to get funding because people can put you in a pigeonhole and they can understand what you're trying to do. Astrobiology is so broad, it's sometimes difficult for people to understand what we're really doing because we ask these very broad questions. But that's also its strength. That's one of the most exciting things about astrobiology is it cuts across disciplinary boundaries, and that's where some of the most exciting science is. So its challenge, I think, ahead, and this is a challenge I see at Edinburgh, and it's, I don't think, a unique challenge, is trying to consolidate around particular questions. In my case, the study of life in extremes. Other people have other questions in astrobiology. But to try and keep that momentum going of creating new jobs, new funding that can sustain a new generation. A really sad thing to see is lots of people coming up in astrobiology and not sufficient jobs to give them permanent positions in universities. I mean, that's not just unique to astrobiology. It's true of a lot of science. But in astrobiology, we also have to confront that challenge of trying to keep the momentum of the field going. I would say to anyone listening who's an early career researcher, don't forget astrobiology. If you go into a university and get a conventional job in something like microbiology and astronomy, do really make an effort to set up astrobiology undergraduate lecture courses. They're incredibly popular amongst students, but what that also does is it'll keep the momentum and critical mass in astrobiology going globally by keeping it healthy in universities. So I think that's a really important way of addressing the challenge of critical mass and sustainability. Keep that excitement and those courses going in academic institutions.
Yeah, and here I have to do a little parenthesis and commend the NASA Astrobiology Program and Institute for really being supportive of the Early Career Network, especially through their conference AbGradCon, which has been formative for me and I know for others. Let's see, any other questions do I see online? There's one by Indre, and I think you've tackled this one already a little bit. She was asking, like, if contact was made with ET, what kind of scientists would be the ones making that contact? But I think astrobiologists are the ones who are going to make that contact. Whether it's geologists or biologists or astronomers depends where we find that life. But what are your thoughts?
Yeah, no, I agree. That's what astrobiology is for. It's one of the things. That's one of the things we're supposed to do, is be that connection if that ever happened. Of course, that's a very optimistic thing to happen in the field. We'll be very happy if we find microbes on another planet. That'll give us plenty of work to do. But yes, I mean, thinking hypothetically, if we made contact with intelligent, an intelligent entity, then I think that astrobiologists would play that role, because astrobiologists bring together astronomers, geoscientists, ethicists, all under one subject. So the astrobiology community would be the obvious community to try and find the particular expertise that was needed at the time, whether that's reproducing a signal that might need astronomers with instruments, whether that's deciding on the ethical implications, or if it was a contact that actually happened for real, a being actually landed on the Earth, then you need some biologists and some biochemists to figure out what's going on. So, whatever the situation is, astrobiology, I think, probably has the widest diversity of expertise that could be brought to bear on that challenge of dealing with that situation.
Agreed. There's a great question by Julia. Hi, Julia, who's asking about phosphorus. So as you know, phosphorus is a key element for life on Earth. But how would we get it? How would we find that on the moons in the outer solar system?
Yeah, well, phosphorus detectors is not really my expertise, but there are electrochemical methods of detecting phosphorus in liquids or in rocks. You can also look at particular minerals, for example, using XRD and XRF are two methods, X-ray fluorescence and X-ray diffraction. So X-ray fluorescence will give you the elemental composition of rocks, and that's used to find phosphorus, for example, on Mars. It's been demonstrated in Martian rocks. X-ray diffraction gives you information on particular minerals. So you could find things like apatite, which is a phosphate mineral. If you're looking in liquids like the plumes of Enceladus, then you might want some electrode that will detect phosphorus or phosphate compounds or different oxidation states of phosphorus rather in liquids. So there are an enormous number of chemical and X-ray mineralogical methods of detecting phosphorus in different states in liquid and rocks that will be used in future planetary missions, are used on the surface of Mars to detect phosphate-containing rocks. But that's a good question because, as she rightly points out, phosphorus is one of the CHNOPS elements, and at least for life on Earth is an essential ingredient of things like nucleic acids, other molecules such as adenosine triphosphate, and parts of enzymes as well. So looking for phosphorus is a key part of biochemistry of astrobiology.
And so we're getting close on time, so we have time for one more question. And I see Graham's one up there on biominerals. So you kind of talked about a little bit about detecting biominerals on other worlds. What are your favorite instruments? What would you like to see on a Mars rover?
Okay, so now I'm going to be flippant again. My favorite instrument is a human being. Yeah, I mean, I really, again, you know, I'm so laughing, but there's a serious side to it as well. Very, very difficult to detect truly biominerals. Even on the Earth, when you find minerals in extreme environments, you have to do a lot of work to demonstrate whether they have been produced by biology or whether they are the product of abiotic chemical reactions. And the way around that is to look at the structure of the minerals to see whether they're associated with microfossils, whether they contain isotopic signatures of life, maybe carbon fractionation, negative delta C13 values, and that sort of thing that might tell you that biology has been involved in their formation, whether that's things like carbonates, different iron minerals. Ultimately, to produce a suite of minerals on a rover that will truly detect a biomineral and say that's life is a very, very difficult thing to do. It's difficult on Earth. It's difficult when you're trying to put it on a rover. So I am a true supporter of sending humans to places like Mars with a laboratory and time on their hands, a good year or so on the surface, to do science, to use sophisticated instruments, to be able to go back into the field and collect samples in a real-time way. And you know, unless we stumble across something on Mars using a rover that's unequivocally biological, the best way at least to look at the possibility of biology on Mars over large areas, even if we find life on Mars, to look at the spatial distribution of biominerals and life on Mars, is to send human explorers there in the long term. And as I say, even if we did find evidence of life using a rover, a biomineral, you still can't beat having a human there for a year in a lab, going out and collecting a vast number of samples and looking at the spatial distribution, looking at different types of minerals, collecting fresh samples based on samples they've looked at, say, the day before or the week before. So I am a fan of the human exploration of Mars. Of course, that's not so easy to implement for places like Europa with extreme radiation or Enceladus. So for those sorts of environments, if we want to look for minerals that have interacted with biology, we, at least for the near term, we're limited to robotic missions.
I think if we do find microbes elsewhere, there'll be another big conversation about whether or not it's Earth contaminants before we can make the claim that it's extraterrestrial. Charles, this has been absolutely delightful. Thank you so much for taking the time in your fantastic orange jumpsuit. chatting with us today about your career and your words of wisdom on astrobiology. Any last words of wisdom you want to share?
No, I just want to say thank you for inviting me to take part in this. It's been a pleasure as usual. And, you know, my only last comment is to everyone out there, particularly early career researchers, stick with astrobiology. It's an incredibly exciting field. There's a lot of great stuff that's going to happen over the coming decades. And, you know, wherever you end up in whatever type of scientific career, remain amongst the astrobiology community, because it's a great field to be in. And I encourage you to keep involved and keep contributing. And thanks a lot again for inviting me. It's been great.
Cheers, Charles. Thank you. So listeners, join us next month for Ask an Astrobiologist. Our guest will be Dr. Penny Boston herself. And until then, stay curious and ciao for now.